Hello, I'd like to welcome you to my talk about MACSPlex Cytokine. With my talk, I'd like to show you how to simplify your cytokine measurement and analysis. My talk is divided into 3 main parts and will take around 40 minutes. First, I will give you some details on our MACSPlex Cytokine Kits concerning the principle and the assay overview. Second, I will introduce our MACSQuant Analyzers, the Express Modes, and the setup of a MACSPlex experiment. Third, I will tell you something about the analysis with our MACSPlex Express Modes. And finally, I will give a short summary. After the second and the third part, I will also show the setup of the experiment and the analysis in our software. Okay, let's start. I'd like to start with the principle and assay overview of our MACSPlex Cytokine Kits. What's the purpose of use of our kits? You can use them for the analysis of cytokine levels, for example, in serum or plasma or cell culture supernatant. And you can use the kits for the analysis of cytokine profiles of immune cells, especially of T-cell populations, for example, after in vitro activation. And you can use the results for the detection of T-cell functionality or, for example, for the detection of T-cell differentiation. Up to now, we have released 2 kits. First, the MACSPlex Cytokine 12 Kit Human. That is designed for the detection of 12 soluble cytokines. And those cytokines are IL-2, 4, 5, 6, 9, 10, 12, 17A, GM-CSF, interferon alpha, interferon gamma, and TNF-alpha. And we have also our MACSPlex Cytokine 10 Kit Mouse. And that is designed for the detection of the 10 soluble cytokines IL-2, IL-4, 5, 10, 12, 17A, 23, interferon gamma, TNF-alpha, and GM-CSF. What are the components of our kit? Each kit is designed for 100 tests, and it contains our MACSPlex cytokine capture beads and the detection reagents. Both of them are pre-mixed and ready to use. Then we also have our cytokine standard. This is a cytokine palette of pre-mixed lyophilized cytokines. Then we also have our setup beads to set up the flow cytometer. You don't have to use them when you have a MACSQuant analyzer or a MACSQuant Analyzer 10. And then we also have the buffer, filter plate, and some adhesive foils for the filter plate. There are also some instrument requirements for using the MACSPlex Cytokine Kits. For washing of the plate, you can use a vacuum manifold or a centrifuge that both work for microtiter plates. Then you also need an orbital shaker, and it is also useful, for example, to have a multi-channel pipette to process all the samples. And then for the data acquisition and analysis, you can use our MACSQuant Analyzer or MACSQuant Analyzer 10 in combination with our MACSChill 96 rack, but you can also use other flow cytometers with a blue and a red laser. So now I'd like to come to the principle of the MACSPlex Cytokine Kits. As I mentioned before, one of our components are the MACSPlex capture beads. Those are bead populations that display different fluorescence properties. We call it a so-called color code, and these fluorescence properties are detected in the MACSQuant B1 and the MACSQuant B2 channel. Those are the channels who detect, for example, FITs or PE conjugates as well. So it looks, more or less like this when you look on the right-hand side. Each individual bead population detects a specific cytokine because the capture beads are coupled to anti-cytokine specific antibodies. And when you then incubate your samples or the standards with the capture beads, the cytokines are bound to the specific antibodies on the capture beads. And then to complete this sandwich structure, you incubate with the detection reagent, and the detection reagent are APC conjugated anti-cytokine antibodies. So you have the possibility to detect the signals in the MACSQuant R1 channel, so the APC channel. And the more cytokines are bound to the capture beads, the brighter the APC signal is. Besides the qualitative determination of the analytes, we can also perform a quantitative determination by using standards with known quantities of analytes. I will give you more information on the next slides regarding this. All in all, we can say that we can detect various analytes in only one sample by using our color-coded plex beads. And here I show you a dot plot as an example of our MACSPlex Cytokine 12 Kit human. You see the MACSPlex B1 channel and the MACSPlex B2 channel, and you see in the plot the 12 different bead populations. And I also highlighted the different analytes that are detected by the different bead populations. So what's the experimental overview of our kits? First, you start with the preparation of the standards and the samples and transfer everything to the filter plate. As I mentioned before, your standard is this MACSPlex Cytokine pellet. You add 200 microliter of buffer to reconstitute the cytokine pellet and then get your stock solution with known cytokine concentrations. And out of this stock solution, you perform a serial dilution and get different tubes with defined cytokine concentrations. And those different samples of cytokine concentrations you would transfer to your filter plate and start with column 1 and column 2, and the remaining positions you can fill with your unknown samples you want to analyze. The next step is to add the capture beads to your standard samples and to your unknown samples, incubate everything, then wash the plate, add the MACSPlex buffer and the detection reagent, incubate again, wash the plate again, and then you can resuspend your samples with buffer and start the acquisition on our MACSQuant Analyzer using our Express Modes in the MACSQuantify software. Here I highlighted the real hands-on time you have to really work on the assay. So you see, even if the assay takes some time due to the incubation time, you see that the hands-on time is not so high. So all in all, we can say that the real hands-on time is less than 1 hour. And even the acquisition time of up to 4 hours, you can spend the time with something else in the lab or in the office because the Express Mode or the measurement of the plate on the MACSQuant runs automatically. Over here, although I go into details in the third part of my talk, I'd like to give you also a short overview of the principle of analysis over here. So first, you start with the identification of the singlet beads, then go on with the identification of the MACSPlex populations and assign the analytes to each bead population. And then the third step is to determine the APC medians for each MACSPlex populations. And you have the MACSPlex standard positions, therefore you get the standard values, and based on these standard values, you can determine your cytokine concentrations in your unknown samples. And now I'm going to show you how easy everything is when you use our MACSQuant Express Modes. So now I come to the second part of my talk. I'm going to introduce our MACSQuant Analyzer, our Express Modes, and show you how to set up a MACSPlex experiment. Our MACSQuant flow cytometers are benchtop flow cytometers with up to 10 optical parameters, so forward scatter, side scatter, and up to 8 colors. They can perform multi-sample processing with up to 96 samples, automated sample labeling, automated calibration with beads to have an optimal performance, and they can also do an automated fluorescence compensation and many more nice things. The MACSQuantify software that runs on our devices, the software that runs on our devices is the MACSQuantify software, and our current software is the brand new version 2.6. And all the features I'm going to show you on the next slides are all based on this software 2.6. So what are the general requirements for flow cytometric measurement analysis? So for a successful measurement and analysis, you need an instrument setting. That means voltage settings, gains, perhaps a compensation. Then to visualize everything, you need an analysis template with dot plots and statistics. And then you also need a gating strategy to get your results out of your raw data. Now I want to give you some details regarding the principle of our MACSQuant Express Modes. Those are tools for semi-automated or automated data acquisition and analysis within our MACSQuantify software. We've developed several express modes for several Miltenyi cocktails or kits, and our express modes allow a simplification of flow cytometric data acquisition and analysis by providing predefined experiment settings, predefined acquisition and analysis template, and by application of an automated gating strategy. And this gating strategy is adjusted for each data file. If you already have a MACSQuant analyzer, you have perhaps already used an express mode, or for example, you have used one of our setup express mode like the calibration or our compensation multicolor. Where do you find our express modes? You find them over here under the settings tab, and then you can select the express button and have access to our analysis express mode, where you also find our MACSPlex express modes, and you can also select our setup express modes, for example, calibration or compensation multicolor. So you see, when you use our express modes, already 2 of our 3 requirements are fulfilled. You have the analysis template and you have the gating strategy. Now I will show you how to set up a MACSPlex Cytokine Express Mode experiment. So we have implemented in the software predefined experiments for our MACSPlex experiments. And those can be uploaded automatically when you scan one of our MACSPlex detection barcodes. So for example, the barcode that is on the vial of the capture beads or on the vial of the detection agent. And as soon as you scan it, these predefined experiments are automatically uploaded. We've implemented these experiments for both MACSPlex Cytokine Kits and also for the MACSQuant configurations MACSQuant Analyzer and MACSQuant Analyzer 10. If the scanning of the barcode doesn't work, it isn't an issue because the experiments can also be loaded manually. What are the contents of our predefined experiments? We have set them up according to the setup in the kit datasheet. They use our MACSPlex filter plate as a rack, and they have assigned position for the express modes MACSPlex Standard and MACSPlex Sample. And they have also the auto label for the MACSPlex Cytokine capture beads and the no label selected and assigned. So now perhaps there comes the question, why should I use auto label? I have already stained my whole samples and plates, so why should I use it? There's a reason for this. Because our express modes, MACSPlex Standard and MACSPlex Sample, are used for both MACSPlex Cytokine Kits. And we have to tell the express modes which kit is used at the current experiment, because the express modes need to know how many bead populations they expect for the analysis and which analytes are detected by the bead populations. And therefore, we use this auto label function, because this auto label over here is based on a reagent data bank on the MACSQuant Analyzer. And in this reagent data bank, we've implemented bead information and provide this by using the auto label function. And this bead information, or this whole information, is important for a correct bead identification and assignment of the analytes and a successful acquisition and analysis of your samples. So you see over here, for example, the kit name is shown, and also how many bead populations and what analytes are detected by the beads. And because the whole plate has been already stained, the auto label process is blocked by the selection of the no label. So that's why we have to use the selected and assigned auto label position for the capture beads for successful data acquisition and analysis. So, and if you, or besides providing the bead information, we also have in our new software 2.6 another nice feature using the selected auto label, because in combination with the MACSPlex Express Modes and together with the auto label, the instrument setting is automatically uploaded. So you don't have to care anymore of the instrument setting, because everything is done automatically. The loading and the processing of the setting information we have also implemented in this reagent data bank is done directly before picking of the sample. And it also gives us the possibility to link defined setting information to each kit. So you see, now we have the third requirement for our successful flow cytometric data acquisition and analysis fulfilled. Now we have everything: the instrument setting, the analysis template, and the gating strategy. Here I'd like to show you a short overview of the data acquisition using our Express modes. So you see, the first step is to log into the software. And then the really important part is very short, so you only have to scan the 2D code on the vial of the capture beads, and automatically your predefined experiment is uploaded. Then you have the option to, for example, assign the sample ID and description to your sample positions. Then you verify that everything is correctly defined in the experiment table, and ensure that you have correct liquid levels in all of your MACSPlex filter plate wells. And then the next step is to start the measurement. So you see, it's really, really easy. But this was now a lot of theory, and now I'd like to switch into the software and show it to you. So, okay. Unfortunately, this is not the I only have a PC version of my software over here, so I don't have a MACSQuant Analyzer. Therefore, the scanning of the barcode I cannot perform. But on a MACSQuant Analyzer, you click over here on this barcode icon, and then the barcode reader starts blinking. And you place the vial with the 2D code in front of it, and automatically this barcode is detected and the predefined experiment is uploaded. Because I cannot perform this over here, I do the steps manually. First of all, I see here, due to my annotations, I see that I have a MACSQuant Analyzer configuration over here, because I only have the BioGlue channel and not a BioGreen channel. So now I have a MACSQuant Analyzer, and then I go to Open, Experiment, Public, select MACSQuant Analyzer, and then select the experiment of the kit I've used. For example, the MACSPlex Cytokine 12 Kit Human. And this, what I've done here, is done automatically when you scan the barcode. So you see over here, the MACSPlex Cytokine capture beads and the no label are selected for all positions. Over here, we have the assigned position for the MACSPlex standard. You see that here is a 1 displayed in all the positions. That means we have a group position. So all those 7 standard positions are finally saved within one data file afterwards. and also over here for the second column of the MACSPlex standard. And over here we have the positions for the MACSPlex sample. So there you detect your unknown sample positions. Also, when you use these express modes, predefined experiment settings are uploaded. For example, high flow rate, inactivated mixing of the samples, standard mode, the uptake volume, and the sample volume. So you see, as soon as you have this predefined experiment uploaded, you can go over here on the MACSQuant Analyzer. There you have the Start Measurement button and start your measurement. You find the experiment table to check if everything is correctly defined over here. So you have an overview over the whole plate and see your sample IDs, description, flow rate, etc. The annotation, the selected auto label for all positions, and over here the selected express modes. Here the standard one, and here the sample for the last positions. So you see, it's really easy to define a MACSPlex experiment in our software 2.6. Okay. So, if you don't have a MACSQuant Analyzer, you can set up your flow cytometer using our MACSPlex setup beats. So you can set the device up manually. And on our webpage, we have an application note with general instructions for the MACSPlex assay data acquisition and analysis. You find them under our MACSPlex Cytokine Kits, and then when you go to the library tab, you find it over here. And down here, you have a detailed special protocol when you want to use our MACSPlex Express Modes on our MACSQuant Analyzer. So now I come to my third part and show you some information about the analysis with our MACSPlex Express Modes. So the whole plate has been measured, and now how is the analysis going to be started? On MACSQuant, it is really easy because the Express Mode analysis is automatically applied after finalization of each data file. What means finalization of each data file? I've shown you in the software that, for example, the positions of the MACSPlex standard data are grouped. And I said that all of the 7 positions are saved within one data file. And that means after the last position, the data file is finalized, and also after the last position of these 7 ones, also the analysis is automatically applied. On the analysis computer, you have to start the analysis automatically, and this you perform by opening the submenu of the data file by right-clicking on it, and then select the view with analysis and then the Express Mode name. You see it over here. And automatically, when you select this view with analysis, the Express Mode analysis is started, and that means automatically the predefined analysis template is applied, and also the automated gating strategy is applied to each data file. So, coming back to the principle of analysis, this is a slide I've shown you before, and I now changed it a bit. So again, the principle of the analysis starts with the identification of the singlet beats and the identification of the beat population. This all is done by the Express Mode. The third step is to determine the APC median for each MACSPlex population. And here, the 2 MACSPlex Express Modes, the standard and the sample one, work together. The MACSPlex standard determines the standard values for each analyte. That means it correlates the cytokine concentration versus the APC median, and the MACSPlex Sample Express Mode calculates, based on the standard values, the cytokine concentration for each analyte in your unknown samples. The analysis of the standard and the sample data files is a self-contained system, so later manual modification, for example moving of a region, does not have any effect on the calculated results. So, how does it look on the monitor when you sit in front of your analysis computer? Let's start with the MACSPlex standard analysis. So over here we have the identification of the singlet beats. Here we have the identification of the color-coded bead populations. And over here, the APC median curves are displayed. So it's just showing the rough tendency of the APC medians over your different cytokine standard concentrations. So you see at one glance, okay, I have an increasing behavior of my APC medians, everything looks good. And over here, we display some kind of experiment information, for example, which kit is used. Much more interesting is, of course, the analysis of your MACSPlex sample data files. And also over here, we have the dot plots or the density plots for the identification of the singlet beads and for the identification of the bead populations. Down here, we have a graphic with an overview of the results. So for each analyte, the cytokine concentration is displayed. Over here, we also display the experiment information, but we show a bit more. For example, we also show the rack position of your sample. And down here, there is the really relevant information. There is the cytokine concentration for each analyte displayed. You see that some of the results are marked in red. Those are the not well-defined results. So, if an APC median correlates with only one concentration based on our standard values, the result is well defined and not marked. So this is the area which is dark gray in our theoretical standard curve, which is displayed on this slide. APC medians that can correlate with more than one concentration or that are out of range, for example, of our standard values are marked in red and with exclamation marks. So you see it over here. So we also did a lot in our new software 2.6 to simplify our sample analysis a bit more, and we implemented for our MACSPlex sample analysis a so-called batch analysis. So you have now the option to perform a one-click analysis to analyze your whole experiment. This analysis is based on grouped data files. So that means you can group your sample positions during the measurement, but you can also group, of course, your data files after the measurement. And, of course, still single-file data analysis is possible. So what does that mean? When you select the view with analysis, MACSPlex Sample, in the submenu of your data file, for all positions within this group data file, the analysis is done and the analysis pages are created. So when you group the whole experiment, you have, for example, up to 80 sample positions, and then you have, after your batch analysis, 80 analysis pages in your software, and you can also print them at once. So it is like this. And also a new feature in our software 2.6 is the automated export of the results of the cytokine concentrations and the counts. This is an express mode controlled export that only is done on the analysis computer. As soon as the export is done, you get an information window you see over here. And this tells you where the exported results are saved, so the directory. This is the same directory where also the base MACSQuant data file is saved. And over here you see what data files are written. So you have the exported concentrations and the exported counts, and the name of these exported data files are created out of the file name of the MACSQuant data file and the appendix concentrations or the appendix count. And here we have an excerpt from the concentration export. So we have over here the identifying well ID, sample ID, and description to define or to identify your samples. And then over here we have the analytes and the correlating cytokine concentrations out of your analysis. So again, this was a lot of theory, and now I want to switch again back to the software to show it to you. So I have here two data files. this is a MACSPlex standard data file, and by right-clicking on the data file, I have access to the submenu, and down here I have the I can select View with Analysis MACSPlex Standard, and as soon as I click on it, the analysis is going to start. So now it is ready. You see over here the singlet bead identification. Here we have the red ellipses that identify our different bead populations, and down here we have the APC medians displayed. Down here I have an MACSPlex Sample data file where I have grouped 9 different sample positions after the measurement. So, and now I can also start the analysis by selecting View with Analysis MACSPlex Sample. So, and here we have the export window of our, the information window of our successful exported results. So here we see the directory where my sample data file is saved, and also my exported results are saved down here. And as soon as I click the OK button, the individual analysis pages for each sample position in this group data file are created. And because I have grouped 9 different sample data files, I also get 9 different analysis pages. And by using these buttons, I can click through the pages and see the individual analysis of the sample positions, and I can print them all at once. So, and now I come to the summary of my talk. Our MACSPlex Cytokine Kits and Express Modes allow a simultaneous detection of multiple cytokines in only one sample by using only 3 fluorescent channels on our flow cytometer. By using standards, we can perform a qualitative and quantitative determination of the cytokines, and compared to technologies like, for example, intracellular cytokine staining, we have less hands-on time. When you have a MACSQuant, you can use our MACSPlex Express Modes. There is no additional device necessary. Our MACSPlex Express Modes allow an automated measurement and a simple and rapid automated analysis of samples, including quantification of our cytokines. And you don't need any more additional software for the analysis. You can use our normal MACSQuantify software. So, all Express Mode features I've shown you in my talk are based on our brand new software 2.6. So if you are interested in the software, please contact, for example, your local MACSQuant specialist or one of our technical support teams. So, and now I'm at the end of my talk. I want to thank you for listening, and if you have any questions, I will be pleased to answer them.